Hey guys, I'm going to go through your breadth paper. I'm going to have to film it in a few videos because I'm not sure how big the file can be that I can upload. Okay, obviously pause it at different parts, and if you have any other questions you can email me. So this is the breadth paper. Question number 1, I have already started trying to film this and got interrupted, so ignore the scribbles for now. So, microscopes vary in their magnification and resolution. Which of the rows A to D in the table below is correct? Light microscope, TEM and SEM. You should know that the light microscope has the lowest magnification and resolution, hence my little down arrows. Transmission electron microscope, or TEM, has the highest, hence my little up arrows. And then scanning electron, or SEM, is somewhere in between. So using that basis, we can have a little look. We're going to, it doesn't say maximum, so let's have a look. So a light microscope can have any of these magnifications. Let's use the basis of this one being the highest, this one in between, and this one the lowest. You should have an idea of the different resolutions, okay? But we can use that to work it out. So we would expect the magnification to be the highest one here, and it needs to be higher than both of these. So if we went with this one, 10,000 here and 50,000 here, that would be wrong because that would put the scanning higher than the transmission. Okay, so it's not that one, that's why I've crossed that one out. Next, could be any of these three for magnification because it's higher here than it is here. But then if we look at the resolution, we want a better resolution, so not necessarily higher. So maybe that arrow is not the best thing, but a higher resolution. So that means an even smaller distance between the objects that it can distinguish between. Okay, so we're expecting a better resolution. So 10 is a lot larger than 0.2, so it's not going to be that one. 0.02 is lower than 0.2, so that's a better resolution. 10 and 10, they're the same, so it's not going to be that one. Okay, so we're happy that the magnification's higher, we're happy that the resolution is better. Then if we look over here, like I said, it could be any of these magnifications. Resolution is 200. So if that's something that you'd forgotten, you can work it out if you remember that transmission is better than scanning, scanning is better than light, and so on. So the answer should be C. Okay. Question number 2. Carbs such as starch are made from monosaccharides, monosaccharides joined together. Which of the bonds A to D joins the monosaccharides together? Ester bond, glycosidic, peptide, or phosphodiester? Now, this is just one of those facts that you need to know, and it is a glycosidic bond. Okay, it's just one of those things that you should know. Next one, some inorganic ions have roles in enzyme-controlled reactions. Which of the rows A to D in the table below is correct? Again, this is one of those that you just have to know. Let me just check because I didn't know this one. The answer is D. Okay, the cofactor for amylase is a chlorine ion and a zinc ion. Okay, so that one's D. Just a fact that you've got to know. Fact. This one you can work out if you remember, like I said, that's the best, then that, then that. Okay? So that's all Miss Dixon stuff. So you could put a little RD if you want so that you remember which is which. Next one. Which of the following statements A to D about the nature of the genetic code is incorrect? Okay, so again, this is Miss Dixon's. It is a degenerate code, so that means over time it can, it will not be as efficient, not as good, which we know because as people get older, they start to get wrinkles and things start to break down because the copying of the DNA is not as effective. So that one's true. It's a triplet code. Yes, you should know that. You have 3 bases for every amino acid. It is overlapping. No, definitely not. You have your 3 codes and they do not overlap. So we'll just double check before we come back to that. It is universal. Yep, definitely. So your answer should be C. Ventilation involves various parts of the mammalian respiratory system. Which of the following statements A to D describes inhalation? So this is my stuff. So I would be thinking inhalation, right, that's breathing in. So the rib cage, there's my wonderful rib cage, can you see that? Yeah, is gonna go up and out. Okay, you can do it yourself. You know it goes up and out. The diaphragm, which normally sits like that, is going to contract and push downwards. Okay, so let's have a look. Rib cage moves upwards and outwards. Yes, we've just said that. External intercostal muscles relax. We'll come back to that in a minute. Diaphragm relaxes. No, the relaxed diaphragm goes up like that and the rib cage comes down. Okay, so that is what the relaxed diaphragm looks like. So that is wrong. B, rib cage moves down and in. No, that's when you breathe out, so it's not that. Diaphragm relaxes. Again, no. C, rib cage moves upwards and outwards. External intercostal muscles contract. Diaphragm contracts. That sounds good to me. Yeah. Come back to this one. And finally, rib cage moves down and in. No, that's exhalation. External intercostal muscles contract, diaphragm contracts. That's correct, but the rest are not. So it must be C. Now, external intercostal muscles, they are the muscles on the outside in between your ribs in your rib cage. Okay, so they're going to contract to lift your rib cage up and out. Now just be careful, because I've seen in a question before, they've spoken about internal intercostal muscles, and you should know that muscles come in antagonistic pairs. So if external intercostal muscles contract to lift the rib cage up, the internal intercostal muscles must relax at the same time. Okay, so just really carefully read that bit there. Okay, so it is C. Which of the following A to D are found in prokaryotes and in eukaryotes? This would probably fall under Miss Dixon. A cell wall made of peptidoglycan. No. We know that in plant cells, which are eukaryotes, it's cellulose, and obviously animal cells don't have a cell wall. Circular genomic DNA, no. We know that plants and animals have strands, chromosomes. A nucleus surrounded by a nuclear membrane, yes, that's true for eukaryotes, but prokaryotes do not have a nucleus, so it's not that one. Ribosomes, yes, that's the only thing that they share, because they're really, really, really tiny, so they can fit inside the small prokaryotes and in the eukaryotes. So ribosomes are in both. That's quite a nice, easy one, hopefully. The pressure changes in one mammalian cardiac cycle are shown in the graph below. Which of the following time periods A to D shows ventricular systole? So you should be thinking ventricular obviously means ventricles, so that's going to be a powerful contraction. Systole means contracting. And hopefully you would recognize this graph, and you can see that this here is to do with the ventricles contracting and relaxing. Now, a mistake that a lot of students make is they think, well, it must be from this point to this point, because that's the ventricles contracting. No, if you think about what the graph actually shows, the pressure in the heart going up, that's the ventricles contracting, and then the blood flowing out is then relaxing again as the blood flows out. So it's going to be in between just below 0.2 and about 0.3. Always check your scale as well. So let's have a look. 0 to 0.1, no. 0.2 to 0.3, yeah, it could be. I would have thought it was a bit before 0.2, but all right then. 0.4 to 0.5, no, definitely not. 0.6 to 0.8, no, definitely not. So we'd go with B, even though I'd say it was a bit before that. Don't mind, I'm just being difficult. Okay, next one, can you see that? Yeah. Measles is a serious disease that can be prevented by vaccination. The chart below shows the measles containing vaccine coverage and annual reported cases of measles between 1980 and 2013. So you need to get your head around the graph before you look at the question. Okay, that's important. So here the bars show the number of cases. So we can see from 1980 to 2013, the number of cases has gone down. So number of cases, I'm just going to add a bit of information for myself, decreases over the time period. Can you still see that? There we go. MCV coverage, so that's the measles containing vaccine. So that's how many people or percentage of people that have been vaccinated. So percentage of population vaccinated, put it into simpler language so I can understand it. So we go from 1980, more and more people are being vaccinated. We've got a slight dip here, but we've got lots of people being vaccinated, and then it levels off at around 90%. So increases over the time period. And then if we, it's better if you've got a ruler, obviously. So about 2006 up to about 2006 remains at around 90%. Okay. So the next part asks, let's see if we can get both down there for you. There we go. Which of the following statements, A to D, is a correct interpretation of the chart? An increase in herd immunity resulted in fewer deaths from measles. So you could assume that because more people are getting vaccinated, we've got an increase in herd immunity. However, we don't have any data to show deaths from measles. The only thing we've got is the percentage of people being vaccinated against it and the number of people getting it. The graph doesn't show anything about deaths. So always, like I said, get your head around exactly what it shows first. So it doesn't show this. I mean, that could be the case, but the question is, what is a correct interpretation of the chart? We don't know that from the graph we've got here. The highest number of measles cases occurred when the vaccination coverage was at its lowest. The highest number of cases was when the measles vaccination was at its lowest. Let's have a look. The highest number of cases is here, is when the number of vaccinations is at its lowest. No, its lowest was 1980, and that's not when the highest number of cases was. So that is incorrect. No. A 90% vaccination coverage resulted in fewer than 500,000 cases of measles each year. Let's have a look. So 90% coverage, so we're looking here, we're looking at this bit here, okay, 90% coverage resulted in less than 500,000 cases every year. So the graph is showing us, let's have a look over here, less than 500,000. So this bottom line here shows us 500,000. So is that true? Whenever it is 90%, the cases were less than 500,000. Yes, that is true. Oops. And we'll just double check that we've not made a silly mistake. There's a positive correlation between the number of measles cases and the MCV coverage. Well, that would suggest we would see coverage would go up like that. And cases we would expect to go up like that. Cases. So no, that's definitely not right. So the answer would be C. And that one was, sorry, both of those questions were from my side. Two different fields, G and H, were sampled for 3 common species of wildflower. The results are shown below. We've got daisies, dandelions, and buttercups. Field G, field H. So let's just have a little look what we've got. Okay, that's fairly even. They've both got the same number of different species. So field G has much more evenness than field H, but they both have the same diversity. Sorry, not diversity, same species richness, but different species evenness. Okay, which of the options A to D is correct? Field G will have a greater Simpson's diversity index. Yes, that's true. I'll come back to it, just make sure that we're not making a silly mistake. Field H has greater species evenness. No, definitely not. Field H will have a greater Simpson's diversity index. No. Remember, the Simpson's diversity index takes into account the species richness and the species evenness. They've got the same species richness, but field G is more even, so that means that it's more stable. So that field G will have a higher Simpson's diversity index. So it's looking good so far. Field G has a greater species richness. No. Well, given what they've given us here, we have to assume that they've got the same species richness because they've both got 3 different species there. So your answer should be A. Okay, do one more and then I'll do a separate video. You can go and get a cup of tea or whatever. Which of the following options A to D lists the 3 domains of life? Okay, so remember we have domain, then it goes keep putting clothes on, fat, greasy, kingdom, phylum, class, order, family, genus, species. Domain comes first. Archaea, bacteria, eukaryota. That sounds good. Come back to that. Bacteria, prokaryota, eukaryota. No, because bacteria and prokaryota are kind of the same thing. Bacteria are prokaryotes, but then the prokaryote kingdom got expanded, didn't it? Because there were so many different things. So, nope. Prokaryote, Protoctista and eukaryota, nope. Protoctista, Plantae and Animalia, absolutely not, because these belong to eukaryotes. So archaea, bacteria and eukaryota. You could also be called eubacteria, and it could also be called true bacteria. Okay, so we've got archaea, eukaryota, and then our true bacteria group. So it should be A. Oh, this pen's annoying. It is cool though, look. Ooh. A. Okay, I'm going to stop it there and I'll carry on.